like to welcome you all for this class of mine today. I am Dr. Abhijit Mujumdar, Director and Professor of Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. We will be studying this topic on sterility testing, which is under the subject of pharmaceutical microbiology with a code of BP303T in Dr. APJ Abdul Kalam Technical University, Lucknow. Now, before I begin, we need to have a brief idea of the context, why are we studying this particular topic. Now, in the previous classes, you have seen the importance of this particular pharmaceutical microbiology as far as the quality control division of our industry is concerned. After studying this subject, many of you will be going for pharmaceutical industries, particularly the quality control division, where you have to evaluate the quality of a disinfectant. You have to judge whether a material is properly sterile or not after the product has been prepared. So, sterilization basically it is a process for removing of the microorganisms. That is the process by which microorganisms were removed, that we are calling as sterilization. And in order to be sure that after the process of sterilization has been done, all the microorganisms were killed. In other words, the substance which you have sterilized has been done properly, we carry out this particular test for sterility, which we are calling as sterility testing. To be more specific, sterility testing was done for detecting whether the microorganisms were present or not in the pharmaceutical preparation. This was the objective of sterility testing. Now, let us study what the principle basically lies on, or what is the principle of sterility testing. If the microbes are present in a medium and this medium is providing the nutrition and water kept at a favorable temperature, the surviving organisms will grow and their growth will be measured or estimated by noting the turbidity of the medium. Therefore, what we have to do is, we have to take the medium, one media for aerobic bacteria, other one for anaerobic bacteria, and the third one for fungi. And then after carrying out this test, we have to expose to that particular bacteriological or a fungal media to see whether the organisms have survived. Now, sterility testing is always referred to in the Indian context as Indian pharmacopoeia. And there were two methods of sterility testing. One is membrane filtration technique, which we will be studying in the today's class, and the other one is direct inoculation technique. Now, in membrane filtration technique, the terminology suggests that you have to simply filter the pharmaceutical preparation through a membrane filter, and the filter is such that if any microorganisms are present, that will be retained on the surface of the filter paper, and the solution will be draining through. And if there is any bacteria or fungus which is present in the preparation, they will be retained on the surface of the filter paper, and that filter paper we need to put up in or immersed in the bacteriological media. There are separate media as prescribed by Indian Pharmacopoeia. We will have to put up the filter paper in a bacteriological or a fungal media, and if that media turns turbid, it means there was a presence of contamination, or the bacteria were not removed or fungus were not removed. That is the reason why they have been turning the media turbid, which in turn means the product fails to cross the sterility testing. On the other hand, in direct inoculation, simply take the preparation, inoculate into the media, and you see after incubation whether the media is turbid or not. Now, let us see what are the products which are commonly tested for sterility testing by this membrane filtration technology, or in fact direct inoculation technique. That is, what are the products which are necessary to be sterilized before use? One is injections, 2 implants, 3 syringes, bandages, dressings, needles, surgical instruments, ophthalmic products and parenterals. Before you are sending this product into the market, we need to be sure of that the product is sterile. And in order to ensure that the product is sterile, you have to carry out what we are calling as sterility testing or test for sterility. The only precaution which you need to take during this test is the test mandatorily needs to be carried out in an aseptic environment, so that there is no contamination during the testing procedure. That is, whenever you are doing the test, you have to be very sure of that the media, the area where you are carrying out the test, there is no presence of bacteria. That is, the area, the environment is aseptic. So, this test must be carried out in an aseptic area to avoid any contamination during testing. Let me explain the process. As I have told you in a very brief nutshell, millipore membrane filter made up of esters or a mixture of esters and cellulose with a pore size of 0.45 micrometer and a diameter of 50 millimeter. These two are very important, particularly the pore size. That is, the pore size needs to be restricted to 0.45 micrometer, and the diameter of the filter paper, it should be 50 millimeter. The purpose why we are restricting the pore size to 0.45 micrometer is, if the pore size is more, the bacteria will pass through. That is the reason why we have to restrict the pore size to maximum 0.45 or below, so that if there is a presence of microorganisms, they will be retained on the surface of the filter paper or the surface of the millipore membrane filter paper, to be more specific. Adjust the sterilized filter assembly and filter the solution to be tested for sterility aseptically across the millipore membrane filter. Then, if you are performing in triplicate, transfer one filter paper to and media which supports aerobic bacteria, transfer the filter paper to a media which supports anaerobic bacteria, and the third remaining one you immersed in a media which supports the growth of the fungi. Or if you are carrying out a single process, if you are carrying out a single assembly, transfer the membrane directly by cutting it into two concentric halves. I am repeating once again. Take the filter paper after passing out the solution which you want to test for sterility. You have to cut the filter paper aseptically into two halves. One will be the inner half which you put up in a bacteriological media to be incubated at 37 degrees Celsius, and the other half, that is the outer half, you dip it in a fungal media. The fungal media has to be maintained at 20 to 25 degrees Celsius, and for bacteria we need to maintain at 37 degrees Celsius. A question may come up in your mind, sir, why are we going to immerse the inner half of the filter paper in a bacteriological medium and the outer periphery of the filter paper in a fungal medium? The reason behind this is bacteria are hydrophilic in nature, due to which they try to concentrate near the center of the filter paper. That is the reason why you are cutting the inner half and put it in a bacteriological medium. This is what you try to do. Now, there will be two concentric half. This you put it in a bacteriological medium, the outer half you put it in a fungal medium. That is, after filtration process there will be two halves, one the inner half if you put it in a bacteriological medium, outer half put it in a fungal medium. But the best way of doing this is you prepare in three sets. Set number 1 you put it in a aerobic media, set number 2 anaerobic media, and third one in a fungal media. Now, there are various diluting fluids. Now, these fluids are commonly used. Fluid A, where we dissolve 1 gram of peptic digest of animal tissue or bacteriological peptone in water to make a volume of 1 liter, filter or centrifuge used to remove the suspended solids, pH to be adjusted to 7.1 to plus minus 0.2, dispense in a flask, sterilize in an autoclave at 121.5 degree for a period of 15 minutes. The second fluid which is recommended as by Indian Pharmacopoeia is fluid B or fluid D as per USP. If the test samples containing oil or lecithin, you have to use fluid B. That is, if the test samples contain oil and lecithin, you have to use fluid B. What is fluid B? Fluid B is nothing but fluid A, which we have just now discussed. To it we add 1 ml polysorbate 80, commonly known as 2-in-80. We have to add polysorbate 80, commonly known as 2-in-80. Similarly, adjust the pH to 7.1 plus minus 0.2, dispense in a flask, then sterilize the diluting fluid with the help of autoclave at 121.5 degree for a period of 15 minutes by moist heat sterilization. USP on the other hand refers what is called fluid K. The composition of fluid K is dissolve 5 gram of peptic digest of animal tissue, 3 gram of beef extract, 10 grams of polysorbate 80 in water to make a total volume of 1 liter. Again, pH to be changed to say 6.9 plus minus 2, dispense in a flask, sterilize in autoclave for a period of 15 minutes. Now, the culture media, I have told you one is aerobic media, anaerobic and fungi. Therefore, the media must support the growth even if the minimum number of microorganisms either in bacteria or fungi is present. The media should be such, it should be nutritionally enriched, so that even if one cell is there, that will try to multiply. So, media must support the growth of even a single bacteria if it is present. Therefore, it must have sufficient moisture, adequate pH, adequate nutrition and suitable redox potential. The media which is commonly used, the media which are commonly used for detection of aerobic bacteria is known as fluid thioglycolate media. The media which is commonly used for anaerobic, they are known as alternate fluid thioglycolate media, and the media which is used for detecting fungi is known as soybean casein digest media. Now, let us study what are the composition of the fluid thioglycolate media. It contains L-cysteine, mainly it is a sulfhydryl containing amino acid, sodium chloride to maintain tonicity, dextrose to your energy source, agar as a solidifying agent, yeast extract as a source of carbon, nitrogen and vitamin, sodium thioglycolate 0.5 gram as a reducing agent, pancreatic digest of casein as nutrient, 0.1% as a resazurin dye, mainly to as an oxidation reduction indicator, and water that is mainly used for clear fluid products. Next is alternate thioglycolate media. It also contains L-cysteine in these are the concentrations: sodium chloride 2.5 gram, dextrose 5.5, yeast extract 5 grams, sodium thioglycolate 0.5 gram, pancreatic digest 15 grams, water up to 1 liter, pH 7.1 to 2 plus minus 0.2. It is mainly used for turbid suspension or any viscous product, say cream and so on. And next is the soybean casein digest broth. Their compositions are pancreatic digest of casein, sodium chloride, peptic digest of soybean meal, that is mainly as a source of carbon, nitrogen and essential amino acids. It has having dextrose carbon source and as a reducing agent, dibasic potassium phosphate mainly as a buffer, and pH 7.2 with a water up to 1 liter. Now, there are certain control tubes. So, I hope up to this is clear to all of you, that is we need to simply pass the solution. If it is insoluble, we need to make it solubilized with the help of diluting fluid, which I have told you earlier, pass it across the filter, and then that filter has to be immersed either in a bacteriological medium, or it is in a fungal medium, or it is in a anaerobic medium. Now, apart from this, we have to take some control tubes. Test tube number 1, we take sterile nutrient media. Test tube number 2, we take sterile nutrient media plus organism. Test tube number 3, sterile nutrient media plus the test sample which you want to test. Test tube number 4, sterile nutrient media, test substance plus organism. In all situations, test tube number 1 should be clear. Why? Because test tube number 1, if it is not clear or turbid, it means that the nutrient media has not been properly sterilized. That is the reason why in test tube number 1 should in all cases be absolutely clear. No turbidity should be present. If there is turbidity, it means there is a presence of contamination or media is not properly sterilized. Test tube number 2, media plus organism, it should be always turbid, because the organism has grown, because you have deliberately added any organism. If this media is clear at all in any situation, it means that this nutrient media has not been properly nutritionally enriched. That is, the media has not been properly providing nutrition for the growth of the bacteria. Next, test tube number 3, sterile nutrient media plus test substance. Depending on whether your substance is sterile or it is unsterile, the turbidity or clarity will depend. Whether your substance is sterile or turbid, your turbidity will depend. And next one is sterile nutrient media plus test substance plus organism. What will be the nature in terms of clarity, in terms whether it will be turbid or it will be clear. In all situations, it should be turbid. Why? Because the organism will try to grow in presence of the nutrient media and it will make the media turbid. Now, there are certain situations, say for example, in 1% cases it is not turbid or it is clear. What does it mean? That is, the test substance has an inhibitory effect on the organism, due to which test tube number 4 is clear. You have to compare test tube number 2 and test tube number 4. If test tube number 2 is turbid, it means the media is complete, there is no problem as far as the media is concerned. And if this test tube number 4 is clear, it means, test tube number 2 turbid, test tube number 4 clear. If this is the situation, it means that the test substance is having antimicrobial property, due to which the organisms were inhibited to grow. That is the reason why what you have to do is you have to first deactivate, neutralize the test substance before you are carrying out this test. Now, let us come to the interpolation process, or how to interpret the result. If after this test, after you are putting that membrane filter in a bacteriological or in a fungal medium, there are two situations, whether it will be turbid or it will not be turbid. Turbid means growth is there, non-turbid means clear means there is no growth. So, let me explain in terms of growth and no growth. If after incubation, if there is no growth, the test passes the sterility test. If the media is clear, it means the sample has been properly sterilized, you allow the batch to go and you put up a remark that the batches pass sterility test as per IP. Now, if the growth is observed, you have to keep the containers are to be reserved and a retest has to be done. If there is a growth, you need to do a retest, and during the retest you take the same number of samples. You see during retest whether there is growth or there is no growth. If during the retest, if there is no growth, means allow the batch to pass. If there is no growth during the retest, allow the batch to pass. But if there is a growth observed during retest, try to measure the microorganism. Try to measure the microorganism, and if the microorganism of the retest matches with the original one, the preparation fails to cross the sterility test. If the microorganism vary, you have to do a second retest with double the number of sample. I am repeating once again. If the if there is no growth in the second test, allow the batch to pass through. If there is growth, compare the organism of the first one and the second test. If these two are same, detain. If they are different, allow a second retest. Second retest is done using twice the number of samples. If there is no growth, the preparation passes. If there is growth, the preparation fails to cross the test. Applications in oil, ointment, non-bacteriostatic solid, a soluble powder or liquid having inherent bacteriostatic or fungistatic action. For liquid products, when the volume of the container is 100 ml or more, this particular method is very commonly used. Advantages: wider application for all types of product, a large volume can be tested, applicable for substances in which no effective inactivations are required, subculturing can be eliminated. Disadvantages: possibility of absorption of sufficient medicament on the filter bed, highly skilled staff required, so automatically the cost increases. You will have to refer to Indian Pharmacopoeia and United States Pharmacopoeia. Now, I will be showing you two videos which will have a very clear cut idea about how basically in industry it works on.
All right, and here we are adding the next dilution vial. Once all the solution is added and we turn on the pump, it's going to pump through the filter, collecting the bacteria on the membrane filter for testing. And then the final one. And she pours it in. Okay, this is not the Sartorius system, but it gives you the general idea of how membrane filtration is done. Now, the next step is to check to make sure the valves down here are in the right position. And they look good? Okay, and then we're going to turn on the pump. And how long does this take?
It doesn't take that long.
Okay.
Within seconds.
Okay, so you can see it's being pulled through here, through the manifold, and into the receptacle. Okay? And they are just about done. Okay, so that's done. Now we can take the filters off, and we are going to put them into a Petri dish.
At this point, we want to add 2 mL of the solution that we're testing.
Right. This tube could be an ampule media. Usually it's an ampule. Right here, she's putting the absorbent pad down into the empty Petri dish. This is one way to do it. Another way to do it would be to put it right onto a pre-poured Petri dish. For example, like the universal beer auger plates we make for Miller Coors. In this case, we're going to be saturating the absorbent pad there with the media, and then we're going to be placing the membrane filter onto that pad, and once that's done, we can incubate and then count for colonies. Okay.
Just enough to soak it.
Mm-hmm. And while she's doing that, I'm going to attempt to unscrew this, but I don't think I can do it with one hand.
I am done. So
Okay.
let's do that.
So now we're going to take off one. Let's just do one for the sake of time, because I'm worried about time on my my digital recorder here. And she's going to take off the membrane filter aseptically and add it to the Petri dish. And then...
And that would be how you incubate them. Like that.
upside down.
Upside down.
There you go.
That's why you soak it just enough to soak it. And you can flip it down like that.
It'll stick.
It'll stick, yeah.
Okay.
Gonna work.
Thank you very much. That is your basic lesson in membrane filtration microbiology. Okay, this is your introduction training into membrane filtration, and we're going to be showing you the process. The components are, of course, here is the manifold. This is your pump. This is your receptacle. These are your funnels. And this is the test solution, the Dilution solution we're going to do. So we're starting by putting the membrane filter onto the frit. This is called the frit, and this is an adapter that goes onto the manifold. Then we attach the funnel, to the frit, which is holding the filter, and we do the same for the other two. Aseptically removing the sterile filter, placing it onto the frit, and attaching the funnel. And here the last filter. And the funnel. And lock it in. So now we have the funnel set up on the manifold, and we're going to...
With this, I am coming to the end of our today's class on sterility testing by the membrane filtration technology. In the subsequent classes, we will be studying about how sterility testing can be done in as per Indian pharmacopoeia with the help of direct inoculation. As I have told you that there are two methods, one was membrane filtration by direct inoculation, membrane filtration and direct inoculation technique by which sterility testing is done. So, we will be meeting again in the next class, where we will be studying about the membrane filtration technology by direct inoculation technique. With this, I am coming to the end of my class today. Thank you very much.